Hello, my name is Samantha Hopper. I'm a student at Concordia University, Texas. We study the effects of protein concentration on blowfly development. In forensic science, their main reference for MPMI estimation is blowfly larvae. While many factors have been studied that alter the growth rate of blowflies, such as temperature, humidity, and resource quality, as obesity increases in the United States, it is important to consider if varied body mass indexes affect larval growth. Previous studies done on body tissues that vary in protein concentration, such as brain, muscle, and liver, have resulted in varied growth rates, but could not be isolated to protein or fat percentage. Our research question was, how does varying protein percentage in muscle tissue affect maggot growth rate and body size measurements? We hypothesized two experiments. The first being that as protein percentage increases and population density of the Lucilia serracata decreases, the length of the larvae will tend to increase. While the second, based on natural environment, was that different communities of scavengers may be recruited to baits containing differing protein and fat percentages. The specificity of the environmental factors was important to rule out factors that may alter growth. We used ground meat to avoid difference in nutrition penetrability. The differing organs previously mentioned failed to represent a consistent penetrability. We also added fat to the ground beef because this was the more accurate than commercial representations, as commercial ground beef represents ranges of protein to fat ratios. As fat percentage decreases, the accuracy of the ratio increases. Therefore, the most accurate ground beef representation was 4% fat. We added pure, non-hydrogenated lard to accomplish differing protein percentages. The fat percentages were based on accepted values of underweight, average, and obese individuals to represent varied BMI. The design shows three different factors of larvae density, protein concentration, and time measured. The population density consisted of two levels, 3 larvae and 9 larvae. Protein percentage with 3 levels of high, which was 89% protein, medium, 80% protein, and low, 66% protein. While measurements were taken at 3 times, of 21, 47, and 68 hours after larvae were laid, followed by measurement responses of larvae length and survival. Fly collection took place at Concordia University, where pork liver was used to attract blowflies that were netted, placed in jars, and morphologically identified. Within the jars, female flies were kept until they had laid several hundred eggs, and then preserved for morphological identification under a dissection microscope. The eggs were counted and placed in respective habitats that consisted of small 2-ounce portion containers with equal amounts of shredded coconut fiber for burrowing. Data collection took place at 21, 47, and 68 hours after the eggs were laid, beginning with the killing of maggots using hot water for 30 seconds to prevent swelling or shrinking. Following this, larvae were photographed on gridded microscope slides and preserved for possible future reference in ethanol. The images were examined with a pixel counter and converted to millimeters. This graph shows the length of larvae over 3 days due to protein percentage and population density. Day 1 shows as protein percentage increases from low to high, the length of larvae also increase. While the length due to population density is increased with increased population size of 9 larvae on day 1, it begins to diminish as no significance by day 3. Notice the trend of increased length due to protein percentage can be seen throughout all 3 daily measurements. The outdoor experiment took place at Concordia University, Texas in late October, early November of 2020. The baits consisted of high and low protein percentage with larvae consistent with the larvae growth experiment. The baits were placed in nylon membranes under cages to prevent large scavengers from interrupting. The results from the outdoor conclude that scavenger activity increased throughout the day and peaked in the late afternoon around 4 p.m. while Figure 2 shows the monopolization of baits by the invasive fire ant species, which reduce the abundance of other species recruitment as increased number of fire ants colonize the baits. The results show that protein percentage has lasting effects on larval length of the Lucilia sericata, resulting in increased lengths with increased protein percentages. While population density does not have a lasting effect on larval length, population density does affect early larval length. The outdoor experiment was unsuccessful in observation of the Lucilia sericata. However, it provided useful information to other native scavengers in central Texas in their recruitment to carrion. Species diversity and recruitment was also not found to have any significant effects due to protein percentage. Recruitment did, however, rely on time of day and presence of the fire ant. In conclusion, resource quality was shown to affect the growth of the blowfly larvae and could offer insights into NPMI estimation based on the unintended consequences of dramatic BMIs. Additionally, postmortem interval may be underestimated in forensic situations of morbidly obese individuals. The factors that affect blowfly larval growth need further research and will require the inclusion of other scavenger species such as the fire ant to add realism to the PMI estimates. Thank you sincerely for joining from Samantha Hopper on behalf of Concordia University, Texas.